Here's a statistic that will blow your mind. On October 9th, 2024, the Nobel Prize in Chemistry was awarded to 3 people who solved the problem that had stumped scientists for over 50 years. And they did it using artificial intelligence. Not traditional chemistry, not physics, AI. We're talking about the protein folding problem, a challenge so massive that Cyrus Levinthal calculated it would take longer than the age of the universe to solve by random search. Yet DeepMind's AlphaFold cracked it in a matter of years, not eons. This isn't just a scientific breakthrough. It's the moment biology officially entered the age of artificial intelligence.
Welcome to Mindcast. I'm Will, and today we're diving deep into one of the most consequential scientific achievements of our time, the story of AlphaFold and how it revolutionized our understanding of life itself. Here's my promise to you. By the end of this episode, you'll understand not just what AlphaFold accomplished, but why this represents a fundamental shift in how science works in the 21st century. We'll explore the strategic genius behind DeepMind's approach, unpack the technical evolution from prediction to generation, and confront the very real controversies that have emerged around this Nobel-worthy AI. This isn't just about proteins. It's about the future of discovery itself.
Let's start with our first key insight, the game master's strategy. When Demis Hassabis, CEO of DeepMind, describes their approach to scientific problems, he uses a fascinating analogy. He calls DeepMind the engine room of scientific acceleration and draws a direct line from their conquests in board games, chess and Go, to the physical reality of proteins. Think about this for a moment. In AlphaGo, the AI navigated the search space of board positions using pattern recognition and what we might call artificial intuition. Remember move 37? That was the moment when AlphaGo made a move that no human had ever considered, defying thousands of years of Go wisdom.
In AlphaFold, the AI navigates the energy landscape of a polypeptide chain using the same fundamental approach. But here's what's brilliant about Hassabis' strategy. They didn't choose protein folding randomly. They specifically selected it because it was what he calls a perfect benchmark for artificial general intelligence development. It had a clear objective metric, the distance between predicted and actual atoms. It had a massive data set, the protein databank with thousands of known structures. And crucially, it had a complexity that defied brute force computation.
This is strategic thinking at its finest. They weren't just solving a biology problem, they were using biology to train an AI system that could eventually tackle any complex scientific challenge.
Now, let's dig into the science itself. For over 50 years, scientists had been trying to crack the protein folding problem using 2 main approaches. First was physics-based modeling, essentially trying to simulate all the forces acting on atoms to find the minimum energy state. Think van der Waals forces, electrostatics, all the nitty-gritty physics. The problem, the computational cost scaled exponentially with protein length. The second approach was homology modeling, using evolution as a guide. If your target protein looks similar to one with a known structure, you could map the new sequence onto the old structure. This worked, but only if you had evolutionary relatives. For orphan proteins, those with no known relatives, this approach failed catastrophically. Then came AlphaFold 1 in 2018, and everything changed. Instead of simulating folding or threading sequences, AF1 treated the protein as a geometric object. It predicted a distogram, essentially a probability map of distances between every pair of amino acids. This was revolutionary, but it was just the beginning. AlphaFold 2, released in 2020, was the industrial-grade solution. It achieved a median score of 92.4 across all targets, essentially indistinguishable from experimental error. The organizers of the scientific competition declared the problem solved.
But here's where it gets really interesting. The architecture that made this possible.
AlphaFold 2 introduced something called the evoformer, and this is where biology meets cutting-edge AI in the most elegant way. The evoformer operates on a beautiful principle. Evolutionary conservation implies structural constraint. Here's how to think about it. If two amino acids that are far apart in the sequence consistently mutate together across different species, they're probably close together in the folded structure. They need each other to maintain the protein's function. The evoformer uses an attention mechanism, similar to what powers large language models like ChatGPT, to iteratively ask two questions. Given these evolutionary patterns, which residues are close? And given these likely distances, which evolutionary patterns make sense? It's like having a conversation between evolution and geometry, mediated by artificial intelligence. And the results speak for themselves. Atomic-level accuracy that rivals the best experimental techniques. But then, DeepMind did something that transformed not just protein prediction, but our entire approach to understanding life. They democratized it. In July 2021, they partnered with the European Bioinformatics Institute to release pre-computed predictions for over 200 million proteins, covering nearly every sequence known to science. They transformed structural biology from a scarcity economy to a post-scarcity economy overnight.
Our second key insight takes us to the generative era, from prediction to generation. In May 2024, just months before the Nobel announcement, DeepMind released AlphaFold 3, and this represents a fundamental paradigm shift. While AlphaFold 2 was a specialist in protein structure, AlphaFold 3 was designed as a generalist biomolecular interaction engine. The most significant change? They adopted a diffusion model, the same technology that powers image generators like DALL-E and Midjourney. But instead of generating images from noise, AlphaFold 3 generates biological structures from noise. Think about what this means. During training, the model takes a valid biological structure and progressively adds random noise until it's just a cloud of atoms. Then it learns to reverse this process, to denoise the cloud step by step to recover the original structure. It's literally learning to hallucinate valid biological reality from chaos. This shift from prediction to generation is profound. AlphaFold 2 could tell you what a protein looked like. AlphaFold 3 can show you how proteins, DNA, RNA, and drug molecules interact with each other. It's moved from answering, "What does this look like?" to "How does life work?" The implications are staggering. Researchers are now using AlphaFold 3 to understand how transcription factors bind to specific DNA sequences, how drug molecules fit into enzymatic pockets, and how antibodies recognize their targets, all within a single computational framework.
Let me give you a concrete example of this technology in action. Matt Higgins's lab at Oxford University was working on a malaria vaccine targeting a protein called PFS4845. This protein is critical for what's called transmission blocking. If humans have antibodies against it, mosquitoes feeding on their blood ingest those antibodies, which then neutralize the parasite and prevent disease spread. The problem? PFS4845 is a hydrophobic, flexible protein that's extremely difficult to study experimentally. For years, researchers couldn't get a clear picture of its structure to design an effective vaccine. Enter AlphaFold 2. The model revealed that PFS4845 has a distinct triangular disk shape with 3 domains, and crucially, it showed how these domains relate to each other, information that was previously unknown. Guided by this AI-generated roadmap, the team engineered stable protein fragments that mimicked the native structure but were much easier to manufacture. They mapped antibody binding sites and designed vaccine candidates that are now progressing toward clinical trials.
This is a direct line from AI prediction to potentially life-saving therapy affecting millions of people worldwide. But it's not just vaccines.
The nuclear pore complex, a massive protein gateway that controls what enters and exits cell nuclei, was resolved using AlphaFold predictions as puzzle pieces fitted into experimental density maps. Drug discovery pipelines now routinely use AlphaFold structures for virtual screening of billion compound libraries.
Now for our third key insight, and this one might surprise you: the cracks in the foundation. Despite all the Nobel-worthy success, AlphaFold isn't perfect, and its limitations reveal something profound about the nature of AI in science. The most dramatic example involves something called chirality, molecular handedness. Here's the issue. Natural proteins are almost exclusively made of L amino acids. Think of them as left-handed building blocks. But in drug design, chemists often use D amino acids, the right-handed mirror images, because they're resistant to biological degradation. A recent study found that when AlphaFold 3 tries to predict structures containing D amino acids, it fails catastrophically about 51% of the time. Even when you explicitly tell it, these are D amino acids, it frequently hallucinates them as L amino acids or distorts them into physically impossible geometries.
Why does this happen? Because the model hasn't learned the laws of physics. It's learned the distribution of the training data. The protein data bank contains 99% L amino acids, so when faced with D amino acids, the AI forces its prediction to conform to what it has seen before, ignoring explicit instructions to the contrary. This reveals a fundamental limitation of deep learning in science. These models are pattern recognition engines, not physics simulators. They can be extraordinarily powerful within their training domain, but potentially dangerous outside it.
The scientific community has also wrestled with access and reproducibility. When AlphaFold 3 was initially released, the code wasn't made available, only a web server with restrictions. Over 800 scientists signed an open letter arguing this violated fundamental scientific principles. Critics used a powerful analogy. Providing a server is like renting a microscope to scientists. It's useful, but it prevents them from understanding how the instrument works or modifying it for new experiments. The tension between commercial interests, biosecurity concerns, and open science has created fault lines that the community is still navigating. This isn't just academic controversy, it goes to the heart of how science should work in the age of AI. Should breakthrough discoveries be controlled by corporations, even well-intentioned ones? How do we balance the democratization of powerful tools with the legitimate concerns about misuse? David Baker's lab at the University of Washington offers a contrasting philosophy with their Rosa-Taffold system. They've maintained an open-source approach, releasing code under permissive licenses and actively encouraging community building. Their tools are positioned as public infrastructure for the field, not corporate assets. The question isn't just which approach produces better science, it's which approach produces science that serves humanity best.
So what does all this mean for you and me? Let me synthesize this into 3 concrete takeaways that extend far beyond biology. First, the strategic power of problem selection. DeepMind didn't randomly stumble onto protein folding. They strategically chose it as a training ground for artificial general intelligence. The lesson? When tackling complex challenges, don't just think about the immediate problem. Think about what solving it teaches you about solving other problems. The protein folding challenge had clear metrics, massive data sets, and required the kind of pattern recognition that would transfer to other domains. This strategic thinking, treating each challenge as preparation for the next, is how breakthrough technologies are built.
Second takeaway, the shift from simulation to generation. We've moved from trying to simulate reality from first principles to learning reality from examples and generating new possibilities. This isn't just happening in biology. We see it in language models, image generation, and soon in material science, drug discovery, and beyond. The implications are profound. Instead of building models of how we think the world works, we're building models that learn how the world actually works from data.
These systems can then generate novel solutions that no human expert might have considered. Third, the importance of understanding limitations. The chirality failures in AlphaFold 3 aren't just technical glitches, they're windows into the fundamental nature of these systems. They're pattern recognition engines, not physics simulators. They're powerful within their training domain, but potentially misleading outside it.
This teaches us to be simultaneously optimistic and cautious about AI and science. Yes, these tools can accelerate discovery in unprecedented ways, but they require human expertise to interpret their outputs, validate their predictions, and understand their boundaries. The real revolution isn't that AI is replacing scientists, it's that AI is becoming the most powerful scientific instrument ever created. Like the telescope or the microscope before it, it's extending human capability into realms we couldn't access before. But just as you need to understand optics to use a telescope effectively, you need to understand machine learning to use AI tools effectively. The winners in this new era won't be those who blindly trust AI outputs, but those who develop AI literacy, the ability to work with these systems as sophisticated collaborators. The 2024 Nobel Prize in Chemistry isn't just recognizing a breakthrough in biology. It's marking the moment when AI became a primary engine of scientific discovery. We've entered what the experts call the simulation age, where we can predict and generate the interactions of life with sufficient accuracy to drive real discovery. From drug design to vaccine development, from understanding cellular machinery to engineering new biological functions, the tools that emerged from the protein folding challenge are reshaping how we understand and manipulate the living world. As we wrap up, here's what I want you to remember. October 9th, 2024 will be remembered not just as the day 3 brilliant scientists won Nobel Prizes, but as the day the scientific establishment formally acknowledged that artificial intelligence has become central to how we discover truth about our universe. The story of AlphaFold is really the story of a new kind of science, one where the most powerful questions aren't just what is, but what could be. It's a science that doesn't just describe reality, but generates new possibilities within it. Whether you're a researcher, an entrepreneur, an investor, or just someone curious about the future, the AlphaFold trajectory offers a playbook for how transformative technologies emerge. Strategic problem selection, patient investment in fundamental capabilities, and the wisdom to balance openness with responsibility. Most importantly, it shows us that the most profound breakthroughs often come not from incremental improvements within existing paradigms, but from entirely new ways of asking old questions. If you enjoyed this deep dive, I'd love to hear your thoughts. What implications of the AI revolution in science are you most excited or concerned about? Reach out on social media or leave a review. Your engagement helps more curious minds find these conversations. Next week, we'll be exploring how quantum computing is poised to solve optimization problems that could revolutionize everything from logistics to materials discovery. Until then, keep questioning, keep learning, and remember, the future belongs to those who understand it. This has been Mindcast. I'm Will. Thanks for thinking with me.